Alright, welcome back everyone. So continuing our classes, starting with this chapter, molecular basis of inheritance, we are discussing this chapter. So in the last video lecture, we have discussed nucleic acid, structure of nucleic acid and the types of nucleic acid, right? So now in this video class, we will discuss the structure of DNA along with their important characters. Okay, then we'll come to RNA. So nucleic acid are of two types, right? DNA and RNA. So now we will see DNA structure. Okay. So we are going to talk about in details about DNA. What is the full form of DNA, by the way? I hope you all know the full form of DNA. Let me just write here. Deoxyribonucleic acid. Okay, deoxyribonucleic acid. Deoxy, why it is called a deoxy? Because at the carbon number 2, oxygen get replaced with single H. We will see that. Nucleic, it is the types of nucleic acid, right? Nucleic acid is the types of nucleic acid. Ribo means pentose sugar. Okay, so we're going to see that in details. First of all, we need to know some important terms about DNA. The scientist name. The DNA, the term was first given by a scientist known as Zacharis. Zacharis. So the term DNA was first given by a scientist, Zacharis. Remember name of the scientist also. While the nucleic acid was given by, the name was given by Altman, right? So the term DNA was given by Zacharis. So what is DNA? DNA is a polymer of nucleotide. DNA is a polymer of nucleotide. Nucleotide we have discussed. It is made up of three things: pentose sugar, nitrogenous base and phosphate group, right? Nucleotide. So DNA is nothing but a polymer of nucleotide. That means the composition will be same in case of DNA also as like nucleic acid and nucleotide, right? Because DNA is a types of nucleic acid only. So nucleic acid का जो composition होंगे DNA का भी वही composition होंगे. Nucleotide क्यों बोलते हैं? क्योंकि when any molecule is made up of three things means nitrogenous bases, pentose sugar and phosphate group, that molecule we call it as a nucleotide. Okay. If I call it as a nucleoside, we will know that if it is nucleoside, we will understand that it is made up of two things: pentose sugar and nitrogenous base, right? There will be no phosphate group. Okay, so DNA is a polymer of nucleotide. That means DNA is made up of three things. Okay, so nucleic acid का जो composition होंगे DNA का भी same वही composition रहेंगे. Okay, so the structure we need to see. DNA का structure in details we will see. We will see the structure given by Watson and Crick as well as the molecular structure of DNA also we will see. See, the structure that we follow are given by Watson and Crick. Okay, Watson and Crick called DNA as double helical structure, and we follow that structure, and it is accepted structure which is given by Watson and Crick. Okay. So when we see the structure of DNA, the actual structure of DNA, we see the structure given by Watson and Crick, two scientists. The structure of DNA was given by two scientists named Watson and Crick. C-R-I-C-K. James Watson and Francis Crick. You can directly write Watson and Crick. So the structure that we need to see in details, or we need to understand, is about the structure given by Watson and Crick. Okay, we will see that, but we will also draw the molecular structure also. So Watson and Crick call DNA as double helical structure. Now we will see that in details, the structure of DNA given by Watson and Crick. So these are the basic things about DNA you need to know before going to the details. So remember the name of the scientist who first given the name of DNA, and then remember the composition of DNA. DNA is known as the polymer of nucleotide. That means it is made up of three things. You already know, we have discussed. When we talk about nucleotide, that means nitrogenous bases, phosphate group, and pentose sugar will be there. And it is a types of DNA is a types of nucleic acid. So ultimately the composition of DNA will be the same as nucleic acid. Okay, so the structure we need to see about the DNA is given by Watson and Crick. So we need to see that structure in details. You might have seen already in your school life the structure of DNA, but here we are going to talk about in details. So the structure that we discuss are given by Watson and Crick, and Watson and Crick called DNA as a double helical structure. So we're going to talk about the double helical. What is double helical? We will see now. Double helical structure of DNA by Watson and let me finish it here, Crick. Okay, so we need to study in details about the structure given by Watson and Crick. So we're going to see one by one along with the structure, along with its features. So first of all, Watson and Crick said that DNA is made up of double helix. Okay, double helix. DNA is made up of double helix. That means there will be one strand and again there will be another strand. Okay, so this is the first point given by Watson and Crick, and we need to know. So first point, let me write. I will draw on the right side of it. Okay, DNA is made up of two strands, that is double helical structure. So first point mentioned by Watson and Crick was that DNA is made up of two strands, and hence they call it as a double helical structure. Helical, the word helical means it twists around. Okay, it twists. So I'm going to draw the structure of DNA. So according to Watson and Crick, DNA has two strands, and that two strands they twist around. Okay, they twist around. Let me show you. This is one strand. Let me draw with black ink. Okay, I'm going to make it big. Okay. So this is one strand and there is another strand, and the strands are not parallel. Okay, the strands are helix. We call it as a helical structure. It means they twist around. Just like, for example, you might have seen a spiral stair somewhere in your surrounding or some places. Just like that, the structure just looks like that. The spiral stair. ओके, सीढ़ी होते हैं ना, ऐसा गोल गोल करके तो जाता है, तो उसी तरह डीएनए का स्ट्रक्चर भी उसी तरह देखने को मिलते हैं, ठीक है। बट रिमेंबर दे डू नॉट क्रॉस विथ ईच अदर, ओके, क्रॉस नहीं होते हैं वो दोनों, ओके, क्रॉस नहीं होते, दे आर जस्ट ट्विस्ट अराउंड ईच अदर। So that's why it is called as helical structure. Double means two strand will be there. This is one strand, there will be another strand. Okay. So two strand. Now see in my diagram I cannot show you the 3D structure, so you need to imagine the structure, okay, don't mistaken that the two strand are cross, एक दूसरे से क्रॉस नहीं होते, ऐसा क्रॉस नहीं होते, okay, remember क्रॉस नहीं होते, they are they are twist around each other but did not cross. So try to imagine, okay, try to imagine the structure. क्रॉस नहीं होते बट एक दूसरे के साथ ऐसे राउंड राउंड करके घूम के रहते हैं, ओके, स्पाइरल स्टेयर जैसा जैसे होते हैं उसी तरह। सो यहाँ पे देखने में इट सीम्स लाइक दे आर क्रॉसिंग विद ईच अदर बट एक्चुअली दे डू नॉट क्रॉस विद ईच अदर, दे आर जस्ट ट्विस्ट अराउंड, ओके। सो ट्राई टू इमेजिन द स्ट्रक्चर और यू कैन सी इन YouTube also or in book also it is given. So try to see the 3D structure. हाँ YouTube में देखो नहीं तो Google में देखो एक बार structure को. So two strand है, there is one strand, there is another strand, and two strands are double helical structure. Helical structure. Helical means twist around. Okay, twist around each other. They twist around each other. Just for example, spiral stair. जो सीढ़ी होता है गोल गोल करके घूमते हैं तो सीढ़ी. You might have seen. Just like that, okay, they do not cross, okay, they do not cross but they are around each other. We will see the next structure, how these two strands combine with each other, how they join with each other. We will see that. So first point he mentioned is that DNA is made up of two strands and that is we call it as a double helical structure. You can write that is double helical structure, or let me write, and it is a double helical structure, okay, and it is a, yeah, let me write it here, and it is a double helical structure. So I hope you understood that there are two strands that twist around each other, we call it as a helical structure, but remember they do not cross, they do not make a cross, ऐसा cross करके नहीं रहते, okay, not like this, they are just around each other and they move, they twist around each other, they twist around each other, just like a spiral stair. That's it. Okay, next. The two strand, next, the feature, the characters given by Watson and Crick. So these are the characters that I am writing, are all the characters given by Watson and Crick only, okay? Because these two scientists have given the structure of, actual structure of DNA. Okay, the next features, the two strands are anti-parallel. Okay, the two strands are anti-parallel. What does that mean? This means if this strand here has 5' free. Now when I talk about 5' free, that means 5' में क्या होते हैं, 5' में हमेशा फॉस्फेट ग्रुप फ्री रहते हैं, राइट, 5' 5' में OH ग्रुप फ्री रहते हैं, फॉस्फेट ग्रुप बाइंड होते हैं 5' में, रिमेंबर. सो ये आके this the opposite of this strand is going to have 3', okay, understood? The anti-parallel means if one strand has 5', the end of it, that strand is going to have 3' OH group free. Now remember, okay, the DNA is what, made up of nucleotide only. That means there will be a pentose sugar only, there will be a nitrogenous bases, and there will be phosphate group, right? These three things makes DNA Okay, composition of nucleic acid and DNA will be the same because both are because DNA are types of nucleic acid only. So DNA is made up of three things. When I talk about 5' that means here is going to have OH group free pentose sugar का diagram याद करो. At the 5' of carbon there is a OH group free, right, which binds with the phosphate group. That means यहाँ पे क्या होंगे, phosphate group bind करेंगे यहाँ पे, ठीक है. तो 3' में क्या होते हैं, OH group free होते हैं फिर से. ओके, डीएनए का 3' में OH ग्रुप फ्री होते हैं. ओके, सो दैट OH ग्रुप हैज अ फंक्शन टू बाइंड अनदर मॉलिक्यूल. कोई भी मॉलिक्यूल बाइंड कर सकता है वो. सो हियर दे आर गोइंग टू बाइंड अनदर न्यूक्लियोटाइड और अनदर डीएनए. वी विल टॉक दिस इन डीएनए रेप्लिकेशन. ओके, सो दिस इज द स्ट्रक्चर ऑफ डीएनए. I hope you understood. So just imagine there is a sugar molecule, pentose sugar. So nitrogenous bases will be there. There will be a phosphate group. So imagine. So the two strands are anti-parallel. That means if one strand has 5', the opposite of other strand, इसका जो opposite strand है, वहाँ पे 3' free OH group free होंगे. Okay. This see this वन लाइक स्ट्रक्चर, ये जो ऐसा लिखते हैं, इसका 2D पावर में जो वन जैसे स्ट्रक्चर लिखते हैं हम लोग, इसको हम लोग प्राइम बोलते हैं, PRIME. ओके, सो व्हेन दिस स्ट्रक्चर इज signifies that it is prime, means free, OH group free. Okay, so if one strand has 5', the other strand means the opposite of that strand is going to have 3', and the end of that strand, उसका जो end part है वहाँ पे 5' होंगे, ठीक है, 5'. So I hope you understood. So this is called two strands are anti-parallel. Anti-parallel means if one strand has 5', the other strand is going to have 3'. Okay, if one strand has 5', the end of it that strand is going to have 3'. The opposite strand का 3' होने से उसका end part 5' होंगे. So उसी को anti-parallel बोलते हैं. Okay, understood? So far what we have seen, so DNA is made up of two strand. ये एक strand हो गया और ये another strand, दो strand, two strand, and two strand are helical structure. Helical means they twist around each other but they do not cross. Okay, they twist around each other. वो दोनों strand कैसे bind होके रहते हैं we will see next. They twist around each other, and the two strands are anti-parallel. What does that mean? If one strand is 5', the other strand is going to have 3' and vice versa here. Understood? Okay. The next structure, what we can see, the purines always pairs with pyrimidine. So when I talk about, or when we talk about purines, that means there will be adenine and guanine, right? When we talk about purines, so there will be adenine and guanine. So purines always pairs with pyrimidines. So when we talk about pyrimidine, since we are talking DNA, so what we will take here, pyrimidines का कौन सा example लेंगे? There will be no uracil, ठीक है, uracil RNA में रहता है, DNA में नहीं रहता है uracil. So here we are going to take the example of cytosine and thymine. Understood? So purine always pairs with pyrimidine. Purines always pairs with pyrimidine. So next characters. Adenine, उसी को ले रहे हैं एग्जाम्पल. वी हैव सीन हियर प्यूरिन पेयर करता है पायरिमिडीन के साथ, लेकिन कैसे पेयर करते हैं नाउ वी विल सी. एडेनिन पेयर्स विथ thymine by making डबल बॉन्ड। इसको हम लोग फिर से पढ़ेंगे। दो साइंटिस्ट है जो given some rules. So we will see after this. Adenine always pairs with thymine by making. Okay, we will see these characters in next scientists which have proposed the bonding of purines and pyrimidine. We will see in the next. Okay, for now just remember here purines always pairs with pyrimidine, but adenine किसके साथ पेयर करते हैं, guanine किसके साथ पेयर करते हैं, उसको हम लोग नेक्स्ट टॉपिक में देखेंगे। जो दूसरा साइंटिस्ट फिर से रूल प्रपोज किया था जहाँ पे उन्होंने बताया कि एडेनिन किसके साथ पेयर करते हैं और कैसे पेयर करते हैं, गुआनिन किसके साथ पेयर करते हैं और कैसे पेयर करते हैं, उसको हम लोग देखेंगे नेक्स्ट टॉपिक में। फॉर नाउ हियर इन डबल हेलिकल स्ट्रक्चर वी विल स्टिक विद दिस ओनली, विद दिस कैरेक्टर्स। सो फॉर वी हैव सीन 2 स्ट्रैंड है, एंटी पैरेलल है, उसके बाद एडेनिन हमेशा प्यूरिन के साथ पेयर करते हैं। ओके, सो आई एम गोइंग टू राइट दिस वन। So next we will see what else. I need a space here again. Okay, I need a space. So let's come to next character. So next what we need to know. We need to know that the diameter of these 2 strands. Okay, the diameter, the distance. ये strand और ये स्ट्रैंड दोनों का डिस्टेंस जो डायमीटर है वो कितना है? द डायमीटर ऑफ दिस 2 स्ट्रैंड्स आर अबाउट 20 आर्मस्ट्रॉन। द यूनिट ए नॉट वी कॉल इट इज ए आर्मस्ट्रॉन। यू माइट नो दिस यूनिट, राइट? सो ये दोनों स्ट्रैंड का जो डिस्टेंस है इट वाज गिवन एज 20 आर्मस्ट्रॉन। ओके, सो रिमेंबर द डिस्टेंस बिटवीन और द डायमीटर बिटवीन the 2 strands are 20 armstrong. So remember this value. Okay, entrance point of view में हमेशा ये सब इसमें इनमें से क्वेश्चन पूछा जाता है। So the next characters we need to know is that the diameter between 2 strands are about 20 armstrong. A naught. ओके, सो रिमेंबर ये दोनों का स्ट्रैंड का जो डायमीटर है इट इज़ अबाउट 20 आर्मस्ट्रॉन। वेरी इंपॉर्टेंट, यू कैन नॉट फॉरगेट। सो नेक्स्ट थिंग व्हाट वी नीड टू नो, देखो यहाँ पे हम लोग को 2 ग्रुप देखने को मिलते हैं, मीन्स 2 डिप्रेशन वी फाइंड। स्टार्टिंग फ्रॉम हियर एंड टिल हियर वी कॉल इट एज ए 1 हेलिक्स। ठीक है, 1 राउंड बोलते हैं हम लोग, यहाँ से यहाँ तक इसको 1 राउंड बोलते हैं हम लोग। ओके, हेलिक्स बोलते हैं, 1 हेलिक्स। सो यहाँ पे डिप्रेशन देखने को मिलते हैं। वी फाइंड ए डिप्रेशन हियर, 1 डिप्रेशन हियर एंड अनदर डिप्रेशन हियर। देखो यहाँ पे एक डिप्रेशन देखने को मिलते हैं यहाँ पे और एक डिप्रेशन देखने को मिलते हैं। ओके, व्हेन डीएनए मेक्स 1 राउंड हेलिक्स, जब डीएनए स्ट्रैंड एक राउंड मारते हैं यहाँ से यहाँ तक 1 राउंड, वी कॉल इट एज ए 1 हेलिक्स। वी फाइंड 2 टाइप्स ऑफ डिप्रेशन, 1 हियर एंड अनदर 1 हियर। ओके, सो ये भी सेम ही रहेंगे डिप्रेशन हियर ऑल्सो। ओके, सो एक डिप्रेशन देखने को मिलते हैं ऊपर में और एक डिप्रेशन देखने को मिलते हैं बीच में। व्हेन डीएनए मेक्स 1 राउंड, दिस डिप्रेशन वी कॉल इट एज ए ग्रूव। ओके, ग्रूव बोलते हैं हम लोग इसको। सो दिस डिप्रेशन इज नोन एज माइनर ग्रूव। जो डिप्रेशन स्टार्ट होते हैं व्हेन डीएनए मेक्स 1 राउंड हेलिक्स, 1 हेलिक्स। Okay, 1 helix. The starting depression is called as minor groove. Then the other depression we call it as a major groove. Okay, और ये भी हम लोग का minor groove it's the same, minor groove बोलते हैं। So between 1 helix we find major group and 2 minor group. Okay, one helix means when DNA makes one turn, यहाँ से यहाँ तक, we call it as a helix. That means one turn. So it goes on again, it goes on. Okay, it goes on. The average length of human DNA is about 2.2 meter. Okay, we will see that when we talk about DNA packaging. 2.2 meter hote hai average length. So I have just shown a simple diagram here, but the structure it goes on. Okay, so from here to till here it makes one round. So when DNA makes one round, we find 2 group, minor group and major group. जो बीच में होते हैं उसको major group बोलते हैं. Major group मतलब ज़्यादा depression होते हैं बीच का. Okay, ये दोनों जो थोड़ा slightly less depression देखने को मिलते हैं, इसलिए minor group बोलते हैं. Group मतलब depression, ठीक है? So यहाँ से यहाँ तक. उसके बाद फिर से it goes on. The structure goes on. Okay, so we find next characters. So in double helical structure of DNA, there is 2 groups. I'm writing short form. Okay, double helical को और डबल स्ट्रैंडेड डीएनए को हम लोग ऐसे भी लिख सकते हैं शॉर्ट फॉर्म में, स्मॉल लेटर डी, स्मॉल लेटर एस. इट स्टैंड्स फॉर डबल स्ट्रैंडेड डीएनए. सो इन डबल स्ट्रैंडेड डीएनए देयर इज 2 ग्रुप. फाइन. कौन सा मेजर ग्रुप जी एंड माइनर ग्रुप. I hope you understood major and minor. जो ज़्यादा डिप्रेशन होंगे बीच में उसको मेजर बोलेंगे, जो लेस डिप्रेशन होंगे उसको माइनर ग्रुप बोलते हैं. ठीक है. जब डीएनए वन टर्न मारते हैं, वन सर्कल, वन हेलिक्स. ओके, दिस आर द ग्रुप. नेक्स्ट व्हाट वी नीड टू नो, okay, the distance, the diameter again. Next we need to know the diameter between one helix DNA. जब DNA यहाँ से one turn मारते हैं यहाँ से यहाँ तक, उसका diameter कितना होंगे? So that's we need to know. See the diameter between one helix DNA. When we talk about helix or one turn, it means one turn DNA. Okay, helix, one turn. So when we when we talk about the one turn DNA, we find the diameter about 34 Armstrong. Okay, so the one turn of DNA has how much diameter? 34 Armstrong. So next important characters, we need to know that the diameter between one turn of DNA, one turn means starting from here to till here, the diameter between one turn of DNA has about 34 Armstrong. Okay, so one turn or one helix of DNA has about approximately 34 Armstrong. Next characters, what we need to know. So when DNA makes one turn, inside we have 10 nitrogenous bases we find. When DNA make one complete turn, it covers with 10 nitrogenous bases. That you need to remember. Nitrogenous bases means purines and pyrimidines, right? Purines and pyrimidine. So जब DNA one turn मारते हैं तब कितने nitrogenous bases होते हैं? उसमें 10 nitrogenous bases होते हैं. So just make it, I'm trying to make it simple here. Here is one nitrogenous bases, 2, 3, 4, 5, 6, 7, 8, 9, 10. So when DNA make one complete turn, the diameter is about 34 Armstrong and it contains 10 nitrogenous bases. 10 nitrogenous bases. Nitrogenous bases matlab purines and pyrimidine. And we have already discussed purine always pairs with pyrimidine. So actual में कैसे रहते हैं? So देखो यहाँ में purines होंगे, यहाँ में pyrimidines होंगे. So adenine always pairs with thymine. हम लोग देखेंगे next. यहाँ में एडेनिन है, यहाँ में थायमिन है, थायमिन होंगे. अगर एडेनिन होते हैं तो थायमिन होंगे यहाँ ऑपोजिट में, because adenine always pairs with thymine by making a double bond. We will see in the next topic. Okay, so here ऐसा नहीं दिखा के we can just simply show like that only. Okay, because we are just here discussing the structure of DNA, double helical structure, and thymine always pairs with adenine by making double bond. This is the actual structure. Okay, actual means yeah, but you can just simply show like that. And guanine always pairs with cytosine by making triple bond. ऐसे होते हैं, okay, normally. But when we talk about the structure of DNA, or when we draw the structure of DNA, we can simply show like that. You don't have to show this way also, but yeah, you can show if you want, but then it will take time. So try to make it simple. Simplicity is best, right? So for our concern, we can just show it like that. There is one nitrogenous basis, two nitrogenous bases, three nitrogenous bases, four nitrogenous bases, and five nitrogenous bases. There are 6, 7, 8, 9, and 10 nitrogenous bases are there, and that's how these two strands, they are linked with each other. How? The nitrogenous bases between each other, they make a bond. Okay, they make a bond. So this strand is actually a pentose sugar, and this is nitrogenous base. So in last class we have seen how these two molecules combine together, right, by making a bond with each other. The bond is known as beta glycosidic bond. We have seen already. So for our concern here, we just show it in a simple way like this. Okay. So in one complete turn of DNA, we find how many nitrogenous bases? So we find 10 nitrogenous bases. So we can write it here. In one complete turn of DNA, it contains 10 nitrogenous bases. Okay, remember कितने nitrogenous bases होते हैं जब DNA one turn मारते हैं? So we find 10 nitrogenous bases. Diameter देखो, confuse मत होना. When the question asks what is the diameter of DNA, then this is the diameter of DNA, 20 Armstrong. Now if the question asks what is the diameter of DNA when it makes one complete turn, so this will be the diameter. Okay, so question को अच्छी तरह से देखना and then write your answer. So एंट्रेंस पॉइंट ऑफ व्यू से बहुत इंपॉर्टेंट है ये सब क्वेश्चन हमेशा पूछते हैं. सो नेक्स्ट थिंग व्हाट वी नीड टू नो, सी द डिस्टेंस बिटवीन दिस टू नाइट्रोजनस बेसिस, दोनों नाइट्रोजनस बेसिस का डिस्टेंस कितना होते हैं, दैट्स इज वेरी इंपॉर्टेंट, वी नीड टू नो. सो जितना ये नाइट्रोजनस बेसिस है उन दोनों का डिस्टेंस कितना होते हैं. सो आई एम गोइंग टू शो इट हियर, और लेट मी शो इट हियर. सो ये दोनों का डिस्टेंस कितना होंगे, ये दोनों का डिस्टेंस, दिस मीन्स द डिस्टेंस बिटवीन द टू नाइट्रोजनस बेसिस. So that is very important. The distance between the two nitrogenous bases are about 3.4 Armstrong. Okay, practice करो, practice करने से सब आ जाएगा. So the distance between the two nitrogenous bases are about 3.4 Armstrong. Okay, वैल्यू को याद रखना, ठीक है. So diagram is very important. Diagram को याद करो, अपने आप आ जाएगा ये सब. Only remembering part is the term. ये ग्रुप का नाम याद रखो, वैल्यू का नाम याद रखो, then ultimately everything will come to your mind. Okay, so the distance between the two nitrogenous bases are about 3.4 Armstrong. This value are given by Watson and Crick. Okay, Watson and Crick, James Watson and Francis Crick. All right, so next characters, what we can see. Okay, this is basically we are talking about human DNA. Okay, human DNA. So DNA बैक्टीरिया में भी होते हैं, एनिमल में भी होते हैं, प्लांट्स में भी होते हैं. दिस इज बेसिकली we are talking about human DNA, and this type of DNA are called it as a B type DNA. DNA का types होते हैं, we will see. This type of DNA are called as B type DNA. DNA का भी types होते हैं, A, B, C, D, Z DNA होते हैं. And this particular DNA are called it as a B type DNA, and this is actually a human DNA we are talking about. Okay, human DNA, B type DNA होते हैं. And when they move, when they twist around, they make a left-handed DNA. Okay, the twist in the left side of it, the DNA. That's why we call it as a left-handed DNA. Okay, left-handed DNA. So this type, what we can write the next character of DNA is known as B type DNA, or you can directly write B DNA, and it is a left-handed DNA. So this is important, one of the important characters. Okay, ये जो DNA होते हैं इसको हम लोग B type DNA बोलते हैं, because जब वो सर्कल ऐसा ट्विस्ट करते हैं एक दूसरे के साथ, they twist in the left side. Okay, just remember left side, they twist in the left side of it. Left side से twist करते रहते हैं, not in the right side, left side. So we call it as a left-handed DNA. That's why. So the Watson and Crick, they perform his experiment. When they observe the structure of DNA, they use one technique, एक instrument को use किया गया था, and that instrument is known as, this experiment was done in a X-ray diffraction टेक्निक. इसको एक्स रे डिफ्रेक्शन टेक्निक बोलते हैं, जहाँ पे वाटसन एंड क्रेग ने ये स्ट्रक्चर को विजुअलाइज और एनालाइज किया. सो दिस आर द सम एक्स्ट्रा थिंग, दिस 2 पॉइंट आर एक्स्ट्रा थिंग आई हैव गिवन. ओके, सो दिस टाइप ऑफ डीएनए आर कॉल्ड इट एज ए बी टाइप डीएनए बिकॉज इट मूव्स, इट टर्न्स इन अ लेफ्ट साइड ऑफ इट. ओके, एंड दिस इज एक्चुअली a human DNA that Watson and Crick analyzed. And the experiment that Watson and Crick performed to analyze this structure of DNA was in a X-ray diffraction technique. All right. So these are the overall structure of DNA. All right. So first of all, it was given by Watson and Crick. And what did he say? Watson and Crick. He said that the DNA is made up of 2 strands. So we have 1 strand, the another strand. Okay. And 2 strands, and they are helical structure. Helical means they twist around like a spiral stair. Okay. So this is one point. Next point, the 2 strands are anti-parallel with each other. That means if one strand has 5' prime, the other strand is going to have 3rd prime. Okay, if one strand is 5' prime, the end of that strand is going to have 3rd prime. If the other strand has 3rd prime, the end of that strand is going to have 5th prime. This ऊपर में जो ऐसा दाग है इसको हम लोग प्राइम बोलते हैं. That means there is a OH group free. 5th prime में क्या join होते हैं? 5th prime में हमेशा phosphate group join होते हैं. 3rd prime में अलग molecule join होंगे. Okay. So next characters, what he said, Watson and Crick said that the purines always pairs with pyrimidine. Okay, we will see that. So purines always pairs with pyrimidine. Okay. Next characters, the diameter between the 2 strands of the DNA is about 20 angstrom. Okay, that's it. Next characters, in one complete turn of DNA or one helix of DNA, we find 2 grooves, minor groove and major groove. That's it. What is groove again? The depression of the DNA. Okay, जब ऐसा twist करते हैं ना, जहाँ पे depression होंगे, वहाँ पे इसको groove बोलते हैं. So next characters, we find the distance between the one complete turn of DNA is about 34 angstrom, and one complete turn it contains 10 nitrogenous bases. So यहाँ से यहाँ तक 1 turn बोलते हैं. So in butene we find 10 nitrogenous bases. So and the distance between two consecutive nitrogenous bases, we should say 2 consecutive nitrogenous bases. ऐसा लिखना, ठीक है? The distance between 2 consecutive nitrogenous bases is about 3.4 angstrom. Okay, and it is a B type of DNA, and it is a left-handed DNA, and the experiment was done in a X-ray diffraction technique. Okay, so this topic is very important. Watson and Crick, very important. Exam में हमेशा पूछते हैं, and entrance point of view very important. The values are very important. Okay, so next we will see some rules provided by other scientists based on the structure of DNA.